So welcome everybody and thank you for joining us today for our virtual event. My name is Sébastien Bello, application specialist with Andor for EMEA, and I'll be your host for this presentation. I am joined today by Dr. Etienne Morel, who is going to talk about virus-host molecular crosstalk to ER-mitochondria membrane interface mobilization in innate immunity. Etienne's main interest in biology concern intracellular compartments, membrane dynamics, trafficking, and interconnections. He obtained his PhD in physiology and cell biology at Pierre and Marie Curie University in Paris, and then specialized later on endosomal membrane dynamics at the University of Geneva and then Columbia University. After getting a PI position at INSERM, and he started to study the role of autophagy in alimentary lipid. He joined the cell biology department of Institut Necker in Paris in 2014 to develop projects on basic autophagy in the context of stress sensing. The research goal of this current team is to understand how mammalian cells organelles cooperate with autophagic machinery to respond to stress stimuli. So just a little housekeeping before we get started. You have all joined this presentation in listen-only mode. If you have any question during the presentation, please type them into the question box in the control panel as we go along, and then we will treat them after the talk. Question will be answered at the end of the presentation, in other words. So now, without further ado, I will hand over to Dr. Morel. Thank you. It's your turn.
Okay.
Etienne.
Okay, thank you. So good afternoon, everyone, or good morning or good evening, wherever you are. Thank you, Sebastian, for the nice introduction. And I would like to thank the organizer for giving me the opportunity of to present the work we are developing in the lab right now. So as Sebastian mentioned, my name is Etienne Morel. I'm a group leader in the cell biology department of INEM, Institut Necker Eniofarma Lab, in the Necker campus in Paris, from INSERM and University of Paris. And the title of my talk is From Virus Host Molecular Crosstalk to ER-mitochondria Membrane Interface Mobilization in Innate Immunity. But as you will see later, I will not talk so much about viruses or innate immunity, but I will talk much more about membrane dynamics in this situation. So a small introduction about what we are doing in the lab. So as Sebastian told you, I'm much more specialized in membrane dynamics, membrane interface mobilization during stress situation in eukaryote cells. So we are interested in stress response, in autophagy, in homeostasis regulation, and also in specific organelles biology such as mitochondria, endoplasmic reticulum, primary cilium, or autophagosome. So in this context, we are dedicated to understand how eukaryotic cells organize the several trafficking pathway and, let's say, interconnection in between key function trafficking pathway inside the cell, such as autophagosomal pathway in blue here on the scheme, secretory pathway, including the ER and the Golgi organelles, and of course, the endosomal pathway, which connect the extracellular medium and the plasma membrane with the degradative organelle lysosome. So in the lab, we are mostly focused on how to understand how those trafficking pathways are interconnected one with the other in response to different kind of stresses, such as nutritional stresses, chemical stresses, mechanical stresses. But today, I will talk to you about infectious stress, and more particularly virus-induced infectious stress, and you will see the effect of viral replication inside cells on ER-mitochondria contact site and on mitochondria morphodynamics regulation. So when we started the study, we picked up one of the most virulent pathogen that we are facing right now, which is influenza viruses. So you all know that influenza viruses are right now a major health concern in the world, especially the subgroup influenza A, which is responsible for the major viral pandemics that we know, with high rates of fertility and morbidity. And as we know, currently for the coronavirus pandemics, the influenza pandemics are also responsible for important social and economic burdens in the world. So the goal of the project initially was to decipher whether presence or replication of influenza virus inside the cell enables endomembrane modification. So we wonder whether presence of replicative virus inside the cell could affect organelles such as mitochondria, endosome, Golgi apparatus, endoplasmic reticulum, and autophagosome, which are the main organelle of the autophagic pathway. And all these experiments have been done in human cells, especially human lung cells, and we used several strains of influenza viruses and other flaviviruses, as you will see later. So first, as I told you, we were interested to know whether presence of influenza viruses, especially H1N1 influenza virus, could affect the number, the disposition, the size of given organelles in eukaryotic cells. So here I'm showing you results concerning autophagosome, Golgi, and endosome and endoplasmic reticulum data. And so we use LC3 to label the autophagosomes, GM130 to label the Golgi apparatus, LAMP1 to label late endosome and lysosome, EEA1 to label the early endosome, and we used transfection of 661 beta protein to label the living the endoplasmic reticulum in those cells. And what we saw in this condition in between control cells and cells that has been infected with H1N1 virus is that we have much more autophagosome biogenesis. So you can see on the top left, we have a fragmentation of the Golgi apparatus. You can see in the middle left, and we don't have any effect on the late endosome numbers. Importantly, when you look at the right, we see a decrease in the total number of early endosome as stained by EA1 marker, and we didn't see any striking effect on the ER morphology in infected cells. So I would like to mention that some of these data have been previously suggested or published in other studies, but especially the number of autophagosome and fragmentation of Golgi in virus-infected cells. But the data we obtained on the lowering of the early endosomal number was brand new. But I won't give you any further details about this. But so the most important data we obtained in this study was about mitochondria. Because when we analyzed cells that have been infected by viruses, so you can see here by the viral NP staining in red on this picture, you can see huge differences in the mitochondrial network morphology. So you can see that in the control cells, in those cells, this is A549 human cells, you can see that most of the time the mitochondria are globular, while the infection via H1N1 virus lead to elongation of the mitochondrial network, as you can see by those pictures and by the quantification of this phenotype. So this was also, let's say, confirmed by electron microscopy, where you can see here differences in between mitochondria in control cells or influenza virus infected cells. And this also lead us to determine that the elongated phenotype we were watching in immunofluorescence was not due to mitochondria that were stick together, but that indeed mitochondria were hyperfused in cells infected with the influenza virus. So This is something we were also able to analyze in live imaging using Mito-tracker as a dye to check on living mitochondria in infected cells. And as you can see here on the top level of this picture, you can see that most, if not all, mitochondria in cells that has been recently infected by H1N1 virus are submitted to elongation. Okay, and at the same time, and really importantly in the context of a study, we also determined by electron microscopy that the contact site in between the ER membrane and the mitochondrial membrane were affected in cells that has been infected by H1N1. So you can see here on the electron micrograph the comparison between cells from the control situation or cells infected by the virus. And you can see on the right the quantification of the contact site. So first, you can see a decrease in the total amount of contact site in cells that has been infected with the virus, as well as a strong decrease in the total density of this contact site in between ER membrane and mitochondrial membrane. So our conclusion at this step was that in cells that has been infected with H1N1 virus, we can observe elongation of the mitochondrial network as well as a disturbance of the ER mitochondria contact site. So sorry, I won't give you so much details about this slide, and I will go directly to this one. So at this step, seeing that we are affecting the shape of the mitochondrial network and we are disturbing the contact site, we wonder whether this could be in relationship in terms of mechanistical explanation with the let's say the classical machinery which is responsible for the fission equilibrium of mitochondria in eukaryotic cells. So the first protein we were interested in is DRP1. So as you can see on the scheme in the middle of this slide, DRP1 is a protein which is recruited at sites of ER mitochondria constriction, contact sites, and DRP1 is recruited there to constrict the mitochondria to promote the mitochondrial fission. On the opposite, OPA1 is one of the proteins required for the fusion of adjacent mitochondria. And I mean, both proteins are really important to check when you want to see if you are going into a fusion or fission state in a certain stress situation. So what we saw is that, in fact, in cells that has been infected with influenza virus, you can see on the left of the slide, we saw in a lesser extent recruitment of DRP1 into TOM20 positive mitochondria. So you can see this by immunofluorescence, and you can see also the quantification on the bottom of the slide. So meaning that we have, in fact, less DRP1 recruited to mitochondrial membrane in cells that has been infected by the virus. On the opposite, when you look on the right, you can see that OPA1, the fusion protein, is much more recruited to mitochondria than in a control situation. This is nicely shown by the immunofluorescence data that I'm showing to you, as well as through the quantification of these events. So all this leads to a conclusion that we are in a pro-fusion state, because we have a decrease of DRP1 recruitment at the mitochondria and increased stabilization of OPA1 at the mitochondria. So we believe that the phenotype we're observing in cells that has been infected by the H1N1 virus, meaning the elongated mitochondria, is in fact due to a balance of DRP1 and OPA1 presence at the mitochondria, which lead to a pro-fusion state. So at this step of the study, we were also interested to decipher what was the trigger in the virus for such a mitochondrial morphodynamics alteration. And we focused notably on double-strand RNA from viral origin, because it was shown in many papers that the double-strand RNA is responsible for many intracellular phenotypes when the virus is in this replicative state. So as a good example, it is shown that double-strand RNA transfection, so it's a single-strand transfection of the double-strand RNA from viral origin in cells, is sufficient to promote interferon production and secretion. This is what you can see on the left of the slide. So you can see that as soon as after 6 hours of double-strand RNA transfection, we are able to detect interferon production and secretion by those cells. And interestingly, in the same condition, we were able to decipher that, in fact, double-strand RNA of viral origin is sufficient to promote the alteration of mitochondrial network we were observing with the full virus. So you can see here in the middle image that transfection of double-strand RNA promotes elongation of the mitochondrial network, as we were detecting with the virus. So the conclusion of this the part of the study is that the viral double-strand RNA is able to mimic the influenza virus on the mitochondrial morphology. And you will see it's kind of important for a later part of the study. So at this step, we wondered whether the mitochondrial morphology alteration we were observing in influenza infected cells could be used as a strategy to counteract the viral replication itself. So for this, we initiate a collaboration with a private company which is based in Lyon, in France, and especially with Dr. Benoît Dechassé, which is the key researcher of Eniofarma. And what is doing Eniofarma? So Eniofarma is a company which is working mostly in silico, and who is able to produce with a strong chemical background some molecules which are themselves able to mimic viral peptide. So, and those viral peptide has been determined in silico study from the mapping of viral and human interaction knowledge. So basically what is happening there is that from in silico studies and from all the literature data that is still available, those people are able to map in 3 dimensions every kind of viral protein interaction with every kind of human protein. And then from this, they are able to synthesize first a peptide and then a molecule that is mimicking the peptide. And from this, for years of studies, Eniofarma was able to build up a full library of different kinds of molecules that are supposed to mimic different kinds of viral and human proteins interaction. So at this level, what we did together was to screen most of the promising molecules. And this screening was aimed at several points. First, we wanted these molecules not to alter the cell viability. Of course, we don't want those molecules to kill the cells, but we wanted to have molecules that were able to modify artificially the mitochondrial morphology, just as we were observing for the influenza virus, but without affecting the mitochondrial function. And the cherry on the cake in the study was to decipher whether or not this kind of molecule could have also an implication in blocking, for example, the viral replication. So to make a long story short, many molecules have been synthesized and tested, and one of them, called Mito-C, as you can see here on the below part of the slide, was a really promising candidate that we decided to study further. So I will not show you all the details about the chemical characterization of Mito-C molecular compound, but what I can tell you is that, in fact, we believe that Mito-C, the chemical compound that we tested, was in fact able to target human proteins of the Innate family protein. So there is 3 proteins in this family, which is also called CSD. So CSD1, which is Mito-Innate, CSD2, which is NAF1, and CSD3, which is called MINT. So it's a bit complicated. But so those three proteins have not been so much characterized. But we know that the main function, let's say the only described function of the three proteins that are really close one to the other in terms of structure, where so the main molecular function of those proteins is to transfer a cluster of ferric and sulfur atoms from the mitochondria matrix inside the ER. So this is what is depicted on the scheme on the bottom left of the slide. So you can see that the clusters are taken charge first by MINT, CSD3, then by Mito-C, CSD1, and then NAF1, CSD2. And so in the middle, you can see the position of the clusters in the structure of the 3 CSD protein, the 3 MINT protein. And in fact, the functions of those proteins are not really described. So of course they are important in iron homeostasis, because as I told you, they are important in clusters transport, but they are also required in ROS regulation, of course in mitochondrial bioenergetics, and it has been suggested that it could be important in autophagy regulation. In terms of pathophysiology, it seems that those proteins could be involved in several oncologic situations and some metabolic diseases such as Wolfram syndrome type 2. And importantly, I must precise that 2 members of this family, especially Mito-C and NAF1, have been identified as ER-mitochondria contact site protein. So it was important in our study to identify those proteins as putative targets of the Mito-C compound. So experimentally speaking, when we tested a fluorescent version of Mito-C compound, you can see here on the top of the slide that as soon as after 2 minutes of treatment with a fluorescent version of Mito-C, we observed a perfect colocalization with a fluorescent version of NAF1, of the CSD2 protein. And because we know this protein is present at the ER-mitochondria interface, probably in contact site, we believe that the Mito-C, in fact, was present immediately at the ER-mitochondria vicinity. And this was later confirmed by similar experiment in which we used different kinds of markers, such as mitotracker, to label living mitochondria in cells. So you can see on the bottom left picture, you can see that the fluorescent version of our molecular compound Mito-C was in fact surrounding mitochondria. It was not colocalizing with mitochondria, but it was surrounding living mitochondria. And on the other hand, when we treat cells with fluorescent version of Mito-C, and those cells has been transfected with a 661 beta red version, which is used as a living marker for endoplasmic reticulum, you can see that we are not seeing a total colocalization, but a partial distribution of the fluorescent version of Mito-C with 661 beta, which strongly suggests that indeed the Mito-C compound is at the interface in between ER and mitochondria. So the most important and probably the key message of the study is that what was the effect of Mito-C on the mitochondrial network? Because that was the goal of the study, to get a drug which enable a modification of the mitochondrial network. And this is exactly what we are seeing in this situation. As soon as after 15 minutes of treatment with a Mito-C compound, we see a mitochondrial network fragmentation, as you can see here on the slide, by immunofluorescence, and by the quantification on the right by the fragmented mitochondrial percentage. Moreover, sorry, I miss. Yeah. So, moreover, so this was also confirmed by electron microscopy once again. So you can see here that we have really a change in morphology of the mitochondria when we treat the cells with Mito-C. But on top of that, we also realize that in fact we're also modifying the ER-mitochondria contact site. So, what we observed was an opposite effect than what we observed previously with the virus. And in this situation, we saw that the Mito-C treatment increases the contact site. So you can see on the graph that it increases artificially the number of contact sites and the density of contact sites. So altogether, Mito-C induces a mitochondrial network fragmentation and an increases of the ER-mitochondria tethering events. So of note, we wanted to be sure that the Mito-C compound had not a side effect on the cell viability and the mitochondrial function. So I'm not showing you all the controls, but we have been testing several parameters to assess the absence of side effects. So first, we saw that increasing doses of the Mito-C compound did not induce any cell deaths. Second, we show that the Mito-C treatment has no impact on the mitochondrial respiration. And third, and probably most important, we saw that the effect we observed with the Mito-C treatment on the mitochondrial morphodynamics was reversible. If you look at the data on the bottom of the slide, you can see that a treatment of 15 minutes with Mito-C induce fragmentation of the mitochondria, but a 2 hours washout of the system lead back to a normal situation with, let's say, normal mitochondria. Okay, so everything that we are looking at is not in relationship with cell death or alteration of mitochondrial respiration or functions. So at this step, we wonder how Mito-C could impact on the morphodynamics regulation of mitochondria. So as you remember, Mito-C seems to target the NEAT family proteins, such as NAF1 protein, but it has not been described that those proteins, the NEAT protein, are important in fission or fusion. So what we did is that we looked again to the most important protein in fission of mitochondria, which is DRP1. And here, as you can see, I think quite nicely by immunofluorescence and by quantification, what we saw is that, in fact, the Mito-C treatment, so this is 15 minutes treatment, enables a huge recruitment of DRP1 at the surface of the mitochondria. So this is easy to detect by immunofluorescence, but we detect also this by a biochemical preparation of mitochondrial membranes. So this is what you are seeing by Western blots in the lower part of the slide. And you can see that by comparison of the mitochondrial fraction purified biochemically, you can see that the fraction of DRP1 which is present at the mitochondria in between control cells and cells that has been treated with 15 minutes of Mito-C is really increased in time. So the conclusion of this kind of experiment is that the Mito-C treatment stabilize DRP1 at mitochondrial membranes. And of course, this makes sense with what we observed on the ER-mitochondria contact site. If you remember well, I told you that the Mito-C treatment artificially increases the ER-mitochondria contact site. And because the DRP1 function in the fission of mitochondria is depending on the contact site, it makes sense that, in fact, the Mito-C drug seems to artificially induce ER-mitochondria contact site, and by doing this, probably artificially recruits DRP1 on the site of fission of the mitochondria. Okay, so at this step, what was interesting is that we were facing two different phenotypes. First, we have a drug, a compound, a molecular compound that seems to target the NEAT family protein, which has no deleterious effect on the cell, which promotes really rapidly the fragmentation of the mitochondrial network. On the other end, on the right, you can see that, in fact, we were the first to show that influenza viruses enable elongation of the mitochondrial network. So it's totally different. So the question there was, could we use the Mito-C drug to counteract H1N1 viral infection? And the answer is yes. So if you look at the experimental part of the slide, you can see that when we compare the Mito-C effect on the virus replication by a neuraminidase activity, it's almost comparable with oseltamivir effect, which is a drug known to counteract the influenza viral replication in living cells. So this was shown also with other techniques such as viral nucleoprotein Western blot detection. So you can see on the right that we have a huge decrease in the viral NP protein amount in cells that has been infected with the virus and treated with Mito-C. And this was also confirmed with PFU experiment, so based on cellular lysis induced by the virus. And you can see that increases doses of the Mito-C chemical compound leads to a huge decrease in the viral replication in those situations, in those cells treated with Mito-C chemical compound. So, of course, we wanted to know whether this was due to mitochondrial network morphology. And the answer was yes. So, as you remember, in the first part of my talk, I told you that unexpectedly, we saw that the H1N1 viral replication induces elongation of the mitochondrial network. And so when we check for the mitochondrial network morphology in cells that has been both infected with the virus and treated with the Mito-C chemical compound, you could see that in fact we are back to the normal situation when you compare the control mitochondria and the mitochondria in cells that has been infected and treated with Mito-C. So I think it's clearly visible on the immunofluorescence data as well on the quantification on the right. And it was also confirmed by electron microscopy. On the lower part of the slide, you can see that we are getting from a situation in which you have really long and elongated mitochondria in infected cells, while infected cells that has been treated with Mito-C exhibit globular mitochondria. So all in all, it seems that the Mito-C treatment counteract the mitochondrial phenotype, which was induced by the virus infection, and by the same time is inhibiting the replication of the virus. So, at last but not least, we check for ER-mitochondria contact site once again. And as expected, what we saw here is that, in fact, along with the fact that the viral replication was inhibited and that mitochondria were back to a normal morphology, a globular morphology in those cells, we saw that Mito-C in cells that had been infected by H1N1 virus were much more increased than in the initial situation. So we think that everything here is connected because if Mito-C is able to counteract the virus-induced ER-mitochondria contact site destabilization, then it will explain why the mitochondrial morphology is back to a normal situation. So to go a little bit further, we wonder whether we could use Mito-C not only in influenza virus condition or situation, but in other viruses which were known and which were described to induce mitochondrial elongation. And this is the case of the dengue virus. So here you can see data. So first I would like you to check on the immunofluorescence data on the right. And you can see that in cells that has been infected with dengue virus, you can observe, as we did for influenza virus, you can observe elongated mitochondrial network. So this was shown before. This was published before. But in cells infected with the dengue virus and treated with the Mito-C, we are observing absence of elongation of the mitochondrial network, and this is what is quantified in the lower part of the graph. And at the same time, when we check for the viral replication of dengue virus, we saw that increasing doses of Mito-C inhibit viral replication of dengue virus. So this is another set of experiment, not only in influenza virus, but we show that in fact our chemical compound Mito-C is also able to counteract the dengue virus infection and the mitochondrial alteration which is associated with dengue virus infection. And of note, so in fact, we show that Mito-C enable the blockade of viral replication for H1N1, H3N2, dengue virus, and also SARS-CoV-2 coronavirus. And this is, of course, non-published data. But this is working on viruses which are known to promote mitochondrial hyper-elongation. So to finish, I would like to show you some data about innate immunity. So here we wonder whether the importance of mitochondrial morphodynamics and the requirement of ER-mitochondria contact site could be related to innate immunity. Why? Because it was shown that viruses, intracellular viruses, and of course, much more viral RNA are related to mitochondria and to innate immunity response. So it was basically shown that presence of double-strand viral RNA in the cytoplasm is able, for example, to activate RIG-I. So, and this protein enabled the aggregation of the MAFS protein at the surface of the mitochondria. And this leads to the pro-inflammatory cytokine secretion, such as interferon, and this is occurring through NF-kappa or I-kappa specialized signalization. And what is important here is it has been suggested that this RIG-I pathway, RIG-I-MAFS pathway, in response to double-strand viral RNA, seems to take place locally in space, in a given membrane in between ER and mitochondria. So this makes sense with the data we obtained in the influenza virus story. So we wonder whether we could check for the RIG-I-MAFS pathway in our situation. So to study the RIG-I-MAFS pathway, the best way is to check for interferon production and secretion, because this is how the cells are protecting themselves and are establishing an antiviral response. So, for example, if you look at the graphs on the middle of the slide, you can see that, as expected, presence of the virus leads to production and secretion of interferons, such as gamma-1 or beta, okay? But what was a striking result is that when you treat those infected cells with H1N1 with a Mito-C compound, you could see that the production and secretion of interferon was much more higher in those cells. And interestingly, this was not happening in non-infected cells, meaning that the Mito-C compound itself is not able to promote formation, secretion, synthesis of interferon. You need to activate the RIG-I pathway. So to confirm the dependency on RIG-I and MAF pathway in our system, we decided to inhibit the synthesis of RIG-I on two different cell lines. And so we build up by a CRISPR technique two different cell lines which were knockout for RIG-I. And then in this situation, we tested the viral replication. So first, when you look at the last graph on the bottom on the right, you can see that we check for the viral replication, viral infection via a neuraminidase activity assay, first in control cells. So if you look at the two first bars, you can see that in cells that have been only treated with Mito-C, but still have RIG-I, you can see a nice, let's say, knockdown, lowering of the neuraminidase activity, showing that you are affecting the viral replication in this system. But when you check this neuraminidase activity in cells that has been knocked down for RIG-I, you see that it's much more higher, which makes sense because you don't have any antiviral response anymore. But what was really striking in this experiment is that the antiviral effect of Mito-C that we were observing in infected cell was not present anymore in cells lacking RIG-I. So, and this has been observed in two different kind of cell lines. So basically, the data we obtained here show that the Mito-C, the chemical compound, is able to potentiate the interferon response, which is depending on RIG-I. And this makes sense with the primary hypothesis we had of the innate immunity regulation at the ER-mitochondria interface. So to finish, I would like to propose you a conclusion, a general conclusion. So what we show first, so you have to look on the left part of the slide, we show that in cells that have been infected with influenza, we saw you are naturally a lower interferon response, and we observed elongated mitochondrial network. When we treat those cells with a Mito-C chemical compound, we see a lowering of the viral infection, higher interferon production, and this is also accompanied by a change in the mitochondrial morphodynamics with smaller mitochondria and higher here mitochondria contact site. And we believe, so we can now look on the right of the slide, we believe that this is occurring by altering the NEAT protein, the NAF1 CSD2 protein, because we know that Mito-C is probably altering the structure or the 3D conformation of the protein. So what we believe is that in situation in which you treat the cells with Mito-C, you are probably artificially stabilizing the ER-mitochondria contact site by altering a given function of NAF1. And by doing this, you are promoting an increased recruitment of DRP1 at the surface of the mitochondria, which will lead to the mitochondrial fragmentation we're observing. Okay, and in this situation, it seems that you are, let's say, promoting much more higher production of interferon. So you can see all the details in the two publications we recently get out. The first one last year in EMBO reports, and the last paper about influenza virus in PLOS pathogens that has been recently published a few weeks ago. Okay, so to finish, I would like to give you an overview of the importance of the endoplasmic reticulum dialogue with all the other membrane, meaning how the contact site enable the stress response in eukaryotic cells, especially the ER-mitochondria contact site. So those ER-mitochondria contact sites have been known for years to be really important for mitochondrial morphodynamics, as is notably depicted here by fluorescence. This is really important to regulate the fission and fission events. But not only, it's also important in the calcium signaling. It's also important, as I told you, with the NEAT protein for iron metabolism. It has been required, it has been shown to be required for lipid transfer. It has also been shown essential for autophagous biogenesis, and so on and so on and so on. But what we would like now to draw to your attention is that we must at some point consider the ER-mitochondria contact site as bona fide active platform for the innate immunity and antiviral response. Because what we show is that in fact, presence of virus, or let's say presence of double-strand RNA in the cytoplasm, seems to directly alter the ER-mitochondria contact site's stability. And by doing this, you will affect the mitochondrial morphodynamics, and what we observe is mitochondrial elongation. But probably the most important effect of this alteration of ER-mitochondria contact site is the lowering of the intracellular innate immunity response. And this is for the virus benefit. So what we could say here is that the virus is enabled to target the stability of ER-mitochondria contact site for its own benefit. So this is my last slide. I would like to thank every people in my lab, especially Irene Pilar Castellanos and Diana Molino, who were the two co-first authors of this study. And I would like to mention that this study has been totally made by a full collaboration with Eniofarma in Lyon, notably with Dr. Benoît de Chassé, but also a bunch of labs everywhere in the world, such as Rachel Nussestein, Damien Arnoux, Francesca Giordano, Cédric Delevoye, Yvan Michaelian, Rodrigue Rossignol, and at last but not least, Alicia Ruggieri from Heidelberg and Caroline Goujon of Montpellier University for the help that we get on the virus, influenza virus and dengue virus experiment. I would like to thank you for your attention, and I will be happy to take any question you may have.
Thank you for this great presentation, Etienne. So I put back my camera. So I'm not sure we have a lot of time for questions, but it's a pity because it was really interesting, and I have a lot of questions. I will go quickly. So just a question about super resolution, because indeed I can imagine that there is okay. Someone tell me that it's okay. It will not stop automatically, so we have time. So in terms of resolution, you display that there's some colocalization, co-distribution. Did you try with an increased resolution this type of analysis?
Not really. In fact, I haven't showed every piece of work, but the resolution we obtained with let's say classical confocal microscopy was enough to get information about what we were looking at. So in this specific study, almost everything was done with let's say classical confocal microscopy, but we didn't use super resolution microscopy.
And it was enough, okay.
Yes, it was enough. It was enough, I must say.
Okay, and what about colocalization? Because you used on one side fluorescence microscopy and on the other side electronic microscopy. Would it be interesting to combine both?
You mean like to do correlative microscopy?
Yes, exactly.
Yeah, that will have been awesome. But the thing is that, of course, I mean, you are specialized in this, but every light microscopy technique has its own caveats. But the problem with the correlative microscopy is that the resolution you get with the light microscopy is not so good. But by doing, let's say, classical light microscopy and classical electron microscopy, at least in this study, we get to the point we wanted to reach. Because we are not so much interested in colocalization or co-distribution of partners. We are mostly interested to know whether we have contact site, not contact site, or elongated or morphology of
you don't have to see both exactly on the same snap, on the same image. Yeah.
On this specific study, not really, and no one asked us.
Yes, and what about the dynamics? Yes, my first question is what about the time scale? After what amount of time you get this effect of the Mito-C molecule?
So as soon as we check, in fact, because we tried. So this was done with a spinning disk live microscopy, classical, and we were not able to manage to do below 2 minutes.
Okay.
In fact, so the data I've been showing you were acquired at 2 minutes just because it was not possible to do it before. So our hypothesis is that this molecule, the Mito-C, is unable, so it's able to cross the membrane. I should have the plasma membrane. I should have mentioned this, but in fact, as soon as it's entering the cell, it's going to the target, and the target is sitting there at the ER-mitochondria interface. So it's immediate.
Interesting. So more in the biology or the pharmacology, the effect of Mito-C on dsRNA, you tested it directly on the double-stranded RNA. Yeah. Yes.
We tested this, and it was abolished. Okay, so basically, it's kind of cool situation because you can use dsRNA transfection to mimic viral infection, which is much less dangerous for postdocs and students. Yes. So what we are doing in the lab when we want to check this, we just have to transfect the double-strand RNA, and we observe exactly the same kind of response, like interferon. And in this situation, Mito-C has exactly the same effect. It's counteracting the effect of double-strand RNA.
No interferon production. Okay.
Absolutely.
So interesting. So another question. Okay, I will read the question that we had at the beginning. So, is Mito-C inducing resistance after several viral infections? Do you have a resistance?
So that's one of the key points of the study, even though it's an in vitro study. The fact is that, and you probably know if you're working on viruses, especially influenza, all the drugs like Tamiflu and all those drugs induce resistance because the virus itself is mutating, and that's why the vaccines and all those the drugs that are developing by different companies are not so efficient on influenza. And probably on COVID. That's another story. But the thing is that by targeting human proteins, it's you don't have this side effect, because what we are targeting here is a human protein, or to be more precise, a human protein interaction with a viral protein in space and time. So in fact, there is no resistance. And we did this kind of experiment by comparison with oseltamivir. And after 3 or 4 passages, the oseltamivir is not active anymore on the virus, because the virus has mutated. And in fact, there is no resistance with Mito-C, because the virus doesn't know. It's not targeting a viral protein. So there is no resistance.
Yeah. And another thing, you talked about this, what is great, I guess, the mimesis library and this company developing this kind of protein. It seems great to me. This is something that probably is used for COVID at this moment.
Yes.
And it provides a lot of cool molecules, I guess.
Yes, absolutely. And I could say it's not secret. I mean, many of the molecules that has been developed by Eniofarma, so that have been developed in this context, I mean, to target mitochondrial morphodynamics, biology, and so on.
Yes.
They are affecting the coronavirus replication. In fact, it's much more efficient than with influenza itself. But so in vitro, it's working. But the problem is that in vivo, the Mito-C is unable to. I mean, it's not dangerous, but it's sticking to protein in the plasma. So far, they are not able to use this as a medication.
Yeah, so, I understand. Yeah.
But it's working in vitro, and it's working pretty well on the coronavirus, I must say.
Okay, this will be my last question, more about the elongated mitochondria network upon COVID. What you can say about this?
It's known. It has been described. It has been described that almost every coronavirus in vitro leads to elongation of mitochondria, just like influenza. And this is not so surprising, because coronaviruses, as far as I know, such as dengue viruses, Zika viruses, influenza viruses, are negative double-strand RNA. And it seems that all those RNA viruses induce mitochondrial elongation. That was new for influenza, but it was known for other viruses, including coronaviruses.
And same effect on the contact sites? This,
no one knows.
Okay. Okay. Still a project for someone.
Probably.
Okay, so thanks a lot, Etienne. It was a great talk, and I invite the people to go and check the video that will be available in the next few days for watching. So on the Andor website, on the Learning Center part of our website. So please be sure to check what is going on. Thanks a lot, Etienne.
Thank you, Sebastian, and thank you all for listening. Thank you.
No problem. Bye. This is the end.